Welcome to the unit on viruses. Throughout this unit, we're going to look at different types of viruses, their structures, and how they're able to replicate. I'm also going to pull in some details about the COVID-19 coronavirus called SARS-CoV-2. That way we can try to tie what you're learning in class a little bit to what you're hearing in the news. In this first video, we're going to look at the overall viral structures. Let's take a look. Viruses are different than any of the other microorganisms we have discussed because they are considered acellular, and you may have noticed that they do not have a branch on the evolutionary tree of life. This means that they are not metabolically active. If a virus was sitting on your desk, it cannot grow or replicate or generate any energy or do any chemical reactions. A viral particle consists only of nucleic acid surrounded by a protein coat. It is more like a genome delivery device than a cell. People often debate if viruses are alive or not, with some virologists arguing on one side and other virologists arguing on the other. So instead, we just call them acellular instead of non-living. Because they don't have any of their own cell structures or metabolism, they must hijack those from a cellular organism, which they do by invading the cell cytoplasm and using your own cellular machinery for their reproduction. This makes them obligate intracellular pathogens. Interestingly, the existence of viruses was discovered all the way back in 1892, when it was shown that tobacco mosaic disease, which is the disease shown here in this plant cell and is causing all of this fading on the leaf, was caused by a pathogen that was smaller than a bacteria. Scientists were able to filter the plant extract through a filter that was so small that it removed all the bacteria. But the extract still caused disease when it was inoculated into a healthy plant. The electron microscope was not invented until the 1930s, so this tiny cause could not be viewed under a microscope, and instead it was described as a biological poison, and in fact the word virus is actually Latin for poison. All viruses are composed of both a nucleic acid core that's surrounded by a protein coat called a capsid. We'll represent the nucleic acid here in red, and that is surrounded by capsid protein shown in purple. While some viruses use standard double-stranded DNA like all other cells, others have very different genomes made up of single-stranded DNA or even RNA in different forms. The nucleic acid genome is protected by this protein coat, which has four main structures that we'll look at. For some viruses, this is their entire structure. Others, however, are also surrounded by a lipid bilayer that's called the viral envelope. These viruses are called enveloped viruses, and those without a lipid layer are called naked viruses. Viruses are classified in multiple ways based on the structure of their capsid, the presence of an envelope, or their host range, which is the type of organisms that they can affect. But most common classification is called the Baltimore model, which is based on the genetic material found housed inside the capsid. This is the most common because viruses with the same type of genome are more evolutionarily related to one another. We'll look at each of the structures in a little more detail. Capsid structures are organized structures meant to protect the genome from degradation and deliver it to a cell's cytoplasm for replication. The proteins that make up the capsid are called capsomer proteins, and a capsid typically contains many connected capsomeres. The four main shapes that a capsid may take on are a icosahedral, which forms a polyhedron with 20 identical symmetrical triangular faces; filamentous, which forms a helical tube around the genome itself. These tubes are flexible and built to be just as long as the genome it is. The third type are called tailed bacteriophages. Bacteriophages are viruses of bacteria that have complex structures that look kind of like these little lunar module landing things. They sit down on the surface of the bacterial cell, and you can see them here in the image. This also shows you a good representation of how much smaller a virus is than a bacterial cell. The fourth classification is called amorphous because they don't really have any set shape that falls into either of the other three. Coronaviruses actually fall into this last category of viruses. While all viruses have a genome of nucleic acid and a protein capsid, only some contain a viral envelope. The envelope is a phospholipid bilayer that actually comes from the infected cell itself. We'll see how the virus picks this up when we look at viral replication. Enveloped viruses can have more variation on their surface because the membrane contains embedded membrane proteins that stick out from the surface. These are the spike proteins that you've heard talked about in COVID-19 discussions, because coronaviruses are enveloped viruses, which is seen in this picture here. You can actually see the spike proteins that you've heard talked about in the news. The red in this image, here and all along the cell, is the lipid bilayer itself. Envelopes are beneficial to viruses because they can protect them from destruction by the immune system. However, the presence of an envelope does make it easier to physically destroy the virus itself. Lipid membranes are made up of a lipid, hydrophobic molecules, just like oil you cook with. Everyone is familiar with dish soap dissolving stuck-on oil from food. Well, it does the same thing to the viral envelope, which is why washing your hands is so beneficial. It not only physically removes the virus, but it also helps dissolve the lipid envelope. Without the envelope, the virus is not able to invade other cells because the envelope has a direct role in viral entry into the cell. Remember that viruses that do not have an envelope we call naked viruses. Packaged inside the capsid is the viral genome. Viral genomes are very small, ranging from some that only have 3 to 5 genes to others with 70 to 100 genes. Compare that to E. coli, which has about 3,100 genes, and humans who have about 22,000 genes. The viral genomes can be so tiny because they don't need to encode any of the enzymes needed for functions like metabolism or cell division or DNA replication because they're going to use the host cell's enzymes after infection. Remember we mentioned that viral genomes can be made up of DNA or RNA. They can also be double-stranded or single-stranded. The type of genetic material is very important to how the virus causes infection because of how cells go from genetic material to produce encoded proteins. If you remember from past classes, that process is called the central dogma. And it functions by going from DNA, which gets transcribed to messenger RNA. Remember that process is called transcription. And that messenger RNA is then translated to a protein. If we put our DNA structure in, remember we've got 5' and 3' ends. We also have 5' and 3' ends on our messenger RNA. For DNA viruses, they don't need to do anything special. Their genome is already made of DNA, and so they will enter the cell, enter our cell's nucleus, and then their DNA will be transcribed to messenger RNA, which will then be translated to viral proteins, the way our cell typically handles DNA and molecular information. For DNA viruses, they don't need to do anything special. Their genome is made of DNA, so the cell knows what to do with that. The viral DNA will typically enter the nucleus of a eukaryotic cell, where it will be transcribed instead of the cell's own DNA, and then the produced messenger RNA will be translated in the cytoplasm by the ribosomes. In bacterial cells, this is even simpler because there's no nucleus to enter first. But RNA genomes are another story. RNA genomes can be positive sense, negative sense, or double-stranded. These RNA viruses all need an enzyme called RNA-dependent RNA polymerase in order to replicate in the host. Everyone always learns that the enzyme RNA polymerase is what completes transcription in the cell. But the full name of this enzyme is actually a DNA-dependent RNA polymerase, which means it uses the DNA as a template to make the RNA. This is different than the term an RNA-dependent RNA polymerase. This RNA-dependent RNA polymerase actually uses RNA as a template to make new RNA. This means the role of the RDRP is to take RNA and replicate it and make more of itself. This is a function that does not exist in any other cells, and those cells do not contain an RDRP enzyme. It has to be encoded in the viral genome. This is because making RNA from RNA is not part of the central dogma. There's no cells that can do this process. It is unique to viruses. There's also a special type of RNA virus called retroviruses, which need a RNA-dependent DNA polymerase, also called a reverse transcriptase, in order to replicate. These enzymes use RNA to make a DNA template. Thus, they go in the opposite direction of transcription, which is why they are called reverse transcriptases. Retroviruses make a DNA copy of their RNA genome when they enter the cell, and that DNA becomes integrated in the host cell's chromosomes, where it is then transcribed as any of the other cell's genes would be. HIV is the most well-known retrovirus. We're going to look at these details on the next slide. We're going to look a little bit more closely at the RNA viruses. So RNA viruses can have a positive sense single-stranded RNA genome, a negative sense single-stranded RNA genome, or a double-stranded RNA genome. What those terms mean, the positive sense single-stranded RNA will run from the 5' to 3' direction. The genome for negative sense goes in the opposite way, 3' to 5', while double-stranded RNA looks similar to double-stranded DNA in that you have two anti-parallel strands. If we go back to the central dogma, remember DNA is transcribed to messenger RNA, and the structure of messenger RNA is to run 5' to 3'. It is this structure that the ribosomes are able to recognize and translate into a protein. So when positive sense single-stranded RNA viruses enter the cell, you should notice that their genome looks the same as the messenger RNA. This means that this genome immediately will actually be translated into viral proteins by the cell's ribosomes. One of those viral proteins is the RDRP enzyme that is then able to come back up and replicate to make lots of copies of the viral genome. However, in either negative sense or double-stranded RNA, that can't happen because if you notice, these do not look like messenger RNA, and our cell has no idea what to do with them. So negative sense RNA and double-stranded RNA do not exist in our cells, which means if those genomes get in, they would actually just sit there until they got degraded and nothing would ever happen, so the virus would never replicate. Instead, for these two types of viruses, along with the genome, they also have to bring that RDRP enzyme packaged into the virus itself with them. So if we look at the virus of those, we would have a capsid, we would have the genome, and we would have that RDRP all within the virus itself. This allows, when they get into the cytoplasm, this RDRP can immediately start replicating and making copies of the viral genome. One of those copies will actually be made 5' to 3', so that step 2 can then be translated into viral proteins. So you should notice that the difference between your positive sense and your negative sense or double-stranded is that steps 1 and 2 are reversed. In positive sense, the first thing that happens is translation, followed by replication, whereas in the negative sense or the double-stranded, the first thing that happened was replication by the RDRP enzyme. The second step was then translation, because now we had a piece of RNA that was in the correct orientation to look like messenger RNA. For retroviruses, they are even different because they bring in RNA that is then converted to double-stranded DNA, which becomes part of the cell's chromosome. Now that you know a little about the overall structure of viruses, let's take a look at a recent computer-generated model of the SARS-CoV-2 virus currently causing the current pandemic. This video shows the virus in great detail. It was built using images from a scanning electron microscope, which were first viewed back in April. And then high-resolution 3D protein models were built on top of the images to provide even greater detail than would usually be seen in an SEM. This was built by a company called Visual Science that actually has a number of really cool computer-generated images and videos like this. If you want to take a look, their website is located in a hyperlink in the title of the slide. Let's take a look at the video. So here you can see the envelope and the spike proteins. And inside we've cut away the envelope. Now we're going to look in the capsid. So the pink is the genome. So you can see these bits of it peeking out, but it's protected by the capsid protein. And all the gray is that lipid envelope that was taken from the cell itself. That lipid envelope, remember, gets embedded by proteins. So we've got these small channels here that allow molecules to travel and through. We also have the big spike proteins that you've heard talked about that are involved with attachment, which we'll talk about in the next video. These little Y-shaped proteins are antibodies that your immune system makes that are meant to bind to the virus itself. The last thing I want to mention is that while viruses are very small, they are not the smallest infectious particles that we know of. Infectious particles is a term used for acellular molecules that can cause infectious diseases. While viruses have at least a nucleic acid genome and a protein coat, viriyoids are infectious RNA with no protein code at all, while prions have no genetic code and are actually just infectious proteins. Prions are actually misfolded proteins that, if they get into the body, will cause your normally folded protein to misfold. This makes clumps of misfolded proteins, leading to plaque development in the brain, the most famous prion disease in mad cow disease.